Today we'll be discussing plasmid replication. Plasmids are small circular extra fragments of DNA, commonly found in bacteria, that are capable of replicating independent of the host genome. Though plasmids are not required for survival of a living organism, but encodes essential genetic determinants that enable an organism to adapt and resist unfavorable conditions for better survival. Rolling circle, Col E1 type, and iteron-containing replicons are the common modes through which plasmid replicates, and each mechanism with unique significance to the organism. Plasmid replication mechanisms. So first we'll start with the rolling circle replication mechanism. Rolling circle replication mechanism is specific to the bacteriophage family M13 and the fertility F factor, which encodes for sex pili formation during recombination by means of conjugation. Fragments smaller than 10 kilobase usually replicate by this replication mechanism. As reported in some gram-positive bacteria, it allows the transfer of single standard replication product at a faster rate to the recipient cell through pilus, as in the case of fertility factor, or to the membrane in the case of phage. The initiation step involves the recognition of double-strand ORI by plasmid and DSO-specific rep proteins. ORI, origin of replication site, and rep proteins, replication proteins. Just look at this diagram. This is single-cell DNA having single-strand origin and double-strand origin. So first step is recognition of this double-strand ORI by plasmid and DSO-specific rep proteins. Two enzymatic activities, nicking and strand closing, accompany the rep protein sequence-specific DNA binding activity. Now, nick is produced in one of the strands by enzyme nickases, creating a 5-phosphate and 3-hydroxyl end. Free 3-hydroxyl will be used by DNA polymerase to make new DNA, pushing the old nicked strand off of the template DNA. Many DSOs contain sequences that promote the formation of hairpin and cruciform structures in the supercoiling dependent fashion. Just look at this diagram. This is single cell DNA, and first step is binding of these rep proteins and amino acid tyrosine. So many DSOs contain the sequence that promote the formation of this hairpin and cruciform structures, right? Binding of rep to the DSO is also known to enhance the cruciform extrusion. To enzymatic activity, as I already told you, nicking and strand closing accompany the rep protein sequence specific DNA binding activity. In many RCR, rolling circle replication plasmids, a nick sequence is located at the loop of the hairpin. Just look at this part. This is the hairpin loop, and here nick is produced. In addition to the DSO's rep binding sequence is required for plasmid replication. Reps are highly conserved among this family of plasmids, and sequence comparisons of reps and DSOs suggest that 100 of RCR plasmids may belong to only a few families. Now, rep protein encoded by RCR plasmids remain covalently attached to the 5' end of the nick site via conserved amino acid tyrosine. This is tyrosine. You can see here rep proteins and tyrosine, and here this is tyrosine. Leading strand replication proceed by extension from the free 3' end of the nicked DNA until a complete round of synthesis leaves the parental portion of the leading strand fully displaced. Cleavage and rejoining reaction at the regenerated nick site, catalyzed by rep protein using another tyrosine, result in a covalently closed circular double-strand DNA that contains the newly synthesized leading strand. The DNA polymerase extends the new strand. It displaces the original nicked strand which rolls off the circular template. This rolling circle movement allows for continuous synthesis of multiple copies of displaced strand. The displaced strand can either be circularized by DNA ligase or it can be cleaved into individual monomeric units, which are then also circularized. The displaced single-stranded DNA is then served as the template for the synthesis of its complementary strand, resulting in a double-stranded DNA molecule. To complete the replication process, the displaced leading strand is subsequently converted to double-strand DNA by using single-strand ORI and solely host-encoded proteins. Now, the second mechanism is Col E1 type replication. Col E1 replication is a negative regulation mechanism which enables the plasmid to control its own copy number by involving RNA type 1, RNA type 2, RM protein, and the plasmid itself. Col E1 replication is initiated by means of RNA-RNA interaction and doesn't rely on replication initiation protein encoded by plasmid to regulate its copy number. RNA type 2 that originates 555 base pairs upstream from the replication origin of Col E1 plasmid is transcribed, which marks the start of Col E1 replication. So initiation of, you can see here, initiation of RNA primer. Then a determined hybrid. A determined hybrid with a DNA strand is formed by a loop enriched in G nucleotide positioned 290 of RNA2 and a C-rich region on the template strand positioned 20 nucleotide upstream from the origin. Several strands and loops are exhibited by this newly formed secondary structure. A DNA-RNA hybrid is recognized by enzyme RNase and dissociate the RNA hybrid to the 3' end of RNA2. The resultant RNA primer is linked to the plasmid with free 3' hydroxyl group. And this RNA enabled replication of DNA to begin by providing DNA polymerase, a specific site to initiate nucleotide synthesis. Consequently, a DNA synthesis is commenced with the leading strand is happening. Next mechanism is iteron-containing replicon. This replicon means iteron. It consists of a gene that encodes rep protein for plasmid replication initiation. Set of direct repeat sequence is called iteron. Adjacent AT rich region and DNA boxes, which is a protein required for bacterial chromosome replication initiation. However, length of adjacent AT rich region and number of iterons and DNA A boxes differ in a replicon. So, iteron contain replication begins with the binding of rep protein to the iteron being organized in same orientation of DNA helix. Just look at this diagram. This is iteron, 5' end and 3' end. So, this iteron contain replication. This replication begin with the binding of rep protein to the iteron, right? Being organized in the same direction of the DNA helix, and by binding to the DNA boxes in the replicon, the rep DNA A DNA assembly. This promotes melting of strand at the nearby AT region, to which host replication factor subsequently gain access and promote leading and lagging strand synthesis in the manner analogous to the initiation of replication at chromosomal region ORI. So first step is rep protein binding, then recruitment of host factors. Rep proteins can also bind to iterin on different plasmid molecules, forming a complex that inhibit further replication. This is known as handcuffing. The binding of rep protein to iterin can be competitive. Therefore, many rep proteins and iterins replication can be inhibited. So, by balancing the binding of rep protein to iterin and the subsequent DNA replication, the plasmid maintains a stable copy number within the cell. Thank you.